Hi everyone, in this video we will be talking about key features of neurons that you should know as a medical student. First thing first, neurons as the cell representatives of nervous tissue can be of three functional types: afferent, interneurons, and efferent. Afferent neurons type 1 are actually sensory, meaning they receive information about something basically from external environment. Second type of neuron is interneuron. This is intermediate neuron between sensory and motor and creates additional junctions in between these two cells. And third neuron, the one that actually passes the result of synaptic transduction, is motor neuron. Basically, this neuron passes information from CNS to skeleton muscle as the result of the information that has been received. Next thing you should know about neurons that they differ depending on their morphology. We have multipolar neurons, bipolar, pseudounipolar, and unipolar. So multipolar neurons predominate in our CNS. They basically have body, many dendrites, and only one axon. Bipolar neurons means that they have body and two separate processes that go in different ways, one axon and one dendrite. This type of neuron predominates in sensory organs. And pseudounipolar neurons means that they have one body, one trunk, one process that comes from this body, and this process will be later separated and divided into axon and dendrite, which also go in different ways. This type of neurons predominates in spinal ganglia. And we also may have unipolar neurons, but these are found only in prenatal stage of neurulation only. As I've said, multipolar neurons predominate in our CNS. So now we can take a look how it actually looks like. Here you have a histological specimen of human brain cortex. And this is the biopsy stained with silver impregnation. And with an arrow, you have the body of multipolar neuron. Actually, this is a giant Betz cells, so giant pyramidal cells. And you can see the triangular shaped soma or the body of the neuron and many dendrites that arise from the cells as well as one axon. Next thing I want you to remember about the neurons is that they have body and it is called soma or perikarion. This is the place where we can find nucleus and a specific system of cytoskeleton made up of neurofilaments. So these are actually intermediate filaments with the specific protein for specific type of neuron, depending whether this will be ependymal cells or astrocytes. Plus, soma is very abundant in Golgi body because we need to pack the neurotransmitters for synaptic communications, which we will be taking a look later on. And we have the axonal hillock. So this is the place of thickening where we know the axon arises. Next thing that I want you to remember about the neurons that they have two different processes, axon and dendrite. Axon, despite what type of functional or morphological type of neuron we are talking about, there is always only one axon, and it is the process that carries the information from the body of the neuron to other neurons or to the effectoring organ, and it starts with axonic hillock. Dendrites may be abundant in number if this is a multipolar neuron, or they can be only one dendrite, but eventually it will branch into several dendritic branches. And this is the process that actually gets the information from external environment in case this is sensory neuron, or it gets information from other neurons around. So how does the diagram from previous slide looks like in a real life settings? Here you have the biopsy from cerebellum, also stained with silver impregnation, and with arrows you can see the pear-shaped like bodies of Purkinje cells and their dendritic trees that extend to the first layer of the cerebellar cortex. Next thing I want you to remember about the neurons is the fact that most of them, whenever they are fully differentiated, are staying in the resting phase of the cell cycle, meaning G0. But importantly, they preserve the ability to move back to the cell cycle into G1, S, and so on and so forth. And this is especially important for us in the clinics of neurodegenerative diseases where we have the potential for regenerations of nervous tissue, but of course it is limited. So how do we communicate in between neurons? We have a specific communicational type of intracellular junction called synapse, and it is present exclusively in nervous tissue. So by synapse, we pass electronic impulse, either neurotransmitters, meaning chemicals, from one neuron to another. And depending what parts of these neurons are communicating, our synapse can be exodendritic, meaning axon from one neuron passes information to the dendrite of second neuron. Synapse may be axosomatic, means axon from one neuron passes information not to the dendrite, but straight to the perikarion or soma of the next neuron. And we have axonic synapse whenever one axon passes information to another axon. This is more rare type, but this is also possible. As for the synapse, I want you to remember three crucial parts of this connection, meaning the presynaptic membrane. This is basically, and more commonly, the axon of the first neuron who is transmitting something to the second neuron. Then in between the, for example, axon of one neuron and dendrite of second neuron, we have a space. So synapses, intracellular junctions where the cells actually do not physically touch, and this space is called intrasynaptic cleft. If we talk about chemical synapse, this will be actually filled with vesicles with neurotransmitters. And where do these neurotransmitters go? This will be received by the postsynaptic membrane, and this can be either dendrite in more classical way or soma axon of the second neuron that will receive the information from the first neuron. The last thing I want you to remember about the neurons is that the speed of the synapses and communication in between neurons and organs and CNS and PNS will be depending whether the nerve fibers are myelinated, and they do have the myelin sheath, or they are non-myelinated, and therefore the speed of the transduction will be slower. But the morphology of myelinated and non-myelinated nerve fibers we will discuss in our next video. Thank you for watching. Don't forget to hit the subscribe, like, and comment button below, and see you in the next videos.